Mitochondrial DNA mutation is a serious cause of many incurable genetic diseases in today's society. They are caused when one or more mutated mitochondrial DNA is paired with the normal mitochondrial DNA and cause heteroplasmy. In 2009, the Oregon National Primate Research Center, or ONPRC, began researching and experimenting with female rhesus macaque monkeys to determine whether or not it was possible to eliminate these diseases. During reproduction, the mitochondrial DNA is inherited from the maternal parent to the offspring. To counteract this, the ONPRC have hypothesized that by taking a karyoplast of a spindle chromosome complex in metaphase stage 2 and transplanting it into the perivitelline space of a healthy donor oocyte, they can counteract the carryover of the mutated mitochondrial DNA. Once the karyoplast has been placed, fusion between the karyoplast and the cytoplasm must then be initiated. To do this, two methods were tested to see which was safest and most successful. The first method was the electroporation method, which was successful within an hour. However, the pulsating from the electroporation caused premature progression from metaphase stage 2 through to anaphase stage 2. The second method was the introduction of the spindle chromosome complex to an extract of the Sendai virus. This was successful within half an hour and was able to prevent progression until after intracytoplasmic sperm rejection could occur. Both methods were then tested in fertilization, and the electroporation method continued premature progression until after the 8-cell blastocyst stage. This rendered the electroporation method as undesirable and unsuccessful for this experiment. The Sendai virus was much more successful and was able to hold a standard rate of fertilization, cleavage and blastocyst through till the end. This can be seen in this table, where the Sendai virus is much closer to the control than the electroporation method was. The successful oocyte was then fertilized in vitro and transplanted into a female rhesus macaque monkey to determine whether or not gene replacement had been successful. In this table you can see the 3 females that were successful in carrying an infant, and the stage that the embryo was in when it was transplanted. On April 24, 2009, the first set of twins to gene replacement had been born, and were followed closely by a single infant on May 4th. Once the infants were determined healthy, a series of experiments were conducted to determine whether mutated mitochondrial DNA had been carried over. All 3 infants were seen to be homoplasmic and had only healthy mitochondrial DNA from the donor oocyte. From this we can conclude that basic mitochondrial gene replacement in rhesus macaque monkeys is a successful way of eliminating mutated mitochondrial DNA carryover. And with more experimentation and research, it is possible to expand this out to other species. Thank you for listening.